In this short video, I'm going to discuss how we can use AutoQuant deconvolution for 2D and 3D datasets in Image Pro. Let's briefly consider why deconvolution is such a useful method. In the wide field microscope, the whole sample is illuminated. So whilst the focal plane is illuminated at the highest intensity, planes above and below the focal plane are also illuminated. And the effect of this is that fluorescence light is emitted from the whole sample. So we get emission from the focal plane and from planes above and below the focal plane, meaning that the image becomes degraded by out of focus light. So how do we solve this problem? There have been two main approaches to solve this. One is with optical solutions, such as the laser scanning confocal microscope, in which optics are used to prevent photons from out of focus planes reaching the detector. The second method has been mathematical solutions, mainly deconvolution. There's another third option, which is a combination of optics and mathematics, but these aren't very common. So let's discuss how AutoQuant deconvolution is used in Image Pro. The deconvolution module is required, and we can check the presence of this by selecting About Image Pro from the Help menu and selecting the Licenses tab. Here you can see that I've got the deconvolution module present. I'm now going to open an image and select AutoQuant deconvolution from the Process tab. And the AutoQuant panel opens. Now the panel takes us through 5 different steps. Let's consider each of them. The first step is to confirm the image metadata. Here you can see the image name and dimensions. We can also see the calibration that's currently being applied, and the lens immersion refractive index and the lens numerical aperture. So calibration, refractive index and numerical aperture are all really important when calculating the PSF. In the next step, we confirm channel metadata. So Image Pro needs to know the modality that was used to acquire each channel and the emission wavelength of each channel. The next step is to choose a starting PSF. Let's just take a moment to consider the importance of the point spread function, or PSF. The point spread function describes the response of an imaging system to a point source or point object. So individual points, when observed in a wide field microscope, get convolved by the PSF to produce a degraded image. So here we can see that these 2 points are displayed smeared in both the z-axis and the x-axis. Now we need to understand the point spread function well in order to be able to perform good deconvolution. Now for 2D datasets in Image Pro, there's a choice of 3 starting PSFs. Firstly, there's a theoretical PSF. The initial point spread function for a theoretical PSF will be generated computationally from the supplied metadata, including the modality, calibration, objective numerical aperture and refractive index, and the emission wavelength of the dye. There's the option of a derived PSF. In this case, the PSF estimate is made from the image data itself. This PSF is not dependent on the metadata fields being correct. And finally, there's a measured PSF. In this case, a dataset is used as the initial point spread function estimate. This will be an image of a single sub-resolution bead collected into the same optical configuration as the sample being processed. The measured PSF is often the hardest one to acquire because it's non-trivial to be able to acquire a field with a single bead in it. The next step is to choose the deconvolution method. And we have 2 to choose from. We can either work with an adaptive PSF, blind deconvolution, in which both the PSF and the image adapt with each iteration, or we can use a fixed PSF in which only the image updates with each iteration. The final step is to choose the deconvolution settings. If you deselect the default setting, you can experiment with different numbers of iterations and different noise levels and change subvoluming settings, but for the purpose of this video, I'm going to use the default settings. When everything's been set correctly, you can click on the Deconvolve button and your deconvolution will proceed. So this is an idealized example in which all the metadata has been loaded with the image. Let's consider what happens when we load an image with missing metadata. ImagePro will display warnings where essential information is missing. So here we see the calibration has not been set for this image. We solve this by clicking on the calibration button, which automatically opens the calibration panel. After selecting the correct calibration and applying it to the image, if I return to the AutoQuant panel, the warning is gone. I'm also seeing warnings here because the refractive index of the immersion lens is missing, as is numerical aperture. So I can select the refractive index from the drop-down menu. So this was an air objective with a numerical aperture of 0.75. So here I'm also seeing warnings in step 2 on the channel metadata. So for one of my channels, I'm missing the emission wavelength. So I'm going to add that. And also the modality for all of the channels is missing. That can be selected from the drop-down menu. Let's execute full deconvolution. So here I'm selecting the derived PSF and clicking on the deconvolve button. And within just seconds, ImagePro will deconvolve my image. A nice trick to be able to compare your raw and deconvolved image is to select the Image tab and the Images panel. In the Images panel, I can click on both my raw and my deconvolved image by holding down the Control key and selecting both. Then I right-click on one of them and select Lock Selected Windows from the context menu. After this, ImagePro displays both images alongside each other with the same zoom and pan settings. So here I'm just stretching the contrast of my deconvolved image a little bit to match my raw image. And from the composite controls, I'm hiding the green channel. I'm also hiding the composite controls completely so we can have more space to look at the images. And you can see that the deconvolved image on the left contains much less out of focus blur than the raw image and contains much sharper details. And here I'm just going to reshow the composite controls and reshow the green channel so we can compare all three channels. Let's move on to consider pass-through channels. And by pass-through channels, I mean channels on which you don't wish to apply the deconvolution algorithm. You just want to pass it through unchanged. To illustrate this, I'm going to use a dataset that I've downloaded from the Cell Image Library. This is image 10384 from Bradner and Withers. And this is an image of hippocampal neurons. They've been immunostained for MAP2, which labels dendrites, shown in red, and PSD95, which labels dendritic spines, shown in green. So here you can see the dataset open in ImagePro, and I have the AutoQuant deconvolution panel open. If I try and deconvolve this with a theoretical PSF, you can see that there's an error being generated in the channel metadata. This is because phase contrast is not one of the supported modalities for a theoretical PSF. I could use a derived PSF and deconvolve without problems, but I'd prefer to use a theoretical PSF in this case, but I don't really wish to deconvolve the phase contrast channel. So all I need to do is deselect the phase checkbox in the channel metadata, and when I hit deconvolve, the phase channel will pass through unprocessed. So here, the deconvolution's in progress, should only take a few seconds. And here we see the deconvolved image generated. I'm going to repeat my trick of selecting both of the images in the Panels tab, right-clicking and locking the images. So I've just stretched the contrast on the deconvolved image to match the raw image. And let's hide the fluorescence channels on both images. So you can see clearly that the phase contrast image has been passed through unprocessed. If we look at the fluorescence channels, they're much crisper on the deconvolved image. We can see the dendritic spines really clearly in the deconvolved image. Let's hide the phase channel from each image set, do the comparison, and yeah, I think it looks really clear how much sharper the deconvolved image looks. So next I'd like to demonstrate deconvolution of 3D datasets. So here in ImagePro I've got a 3D dataset open. So it's a 3-channel dataset with multiple Z planes, and if we scroll through Z, we can see it's a good example of a dataset that's been corrupted by out of focus haze. Now when I select the AutoQuant deconvolution panel, you see I only get 2 choices for a starting PSF for 3D, so no derived PSF option is available. All of my metadata is loaded correctly. I'm selecting an adaptive PSF and clicking on the Deconvolve button. Now you notice I get a little balloon appear with a shortcut to show the task manager, so here I can see the progress. Deconvolution is done in the background, so it's useful to be able to see the progress in the task manager, and you see a progress bar for each of the channels being deconvolved here. Thanks to the GPU processing, Image Pro is able to make quick work of this volume. And when the process finishes, which it will do in just a moment, I'm going to lock my deconvolved and my wide field datasets for comparison. We should be able to scroll through the Z planes and see how they both look. So there we go, that's finished. So just before I lock both, right click on one of them and select the lock option. You can see it's a dramatic improvement in quality of the deconvolved dataset. So all of the out of focus haze is gone. The optical sections look much sharper. And I'm just stretching the contrast, just so it matches the raw data a bit better. And you can actually also lock the datasets using the 3D view, so we can make the comparison of the 3D rendered volumes. And I'm sure you'll agree it's much clearer, it's much more impressive to render the deconvolved dataset. It has much more of an impact in a presentation. The next thing I'd like to demonstrate is processing of multiple images in a folder using the AutoQuant deconvolution batch processing function. So here I've opened one of a folder of images which are all similar, and if I select the AutoQuant deconvolution button, the panel opens and you can see that all my metadata is loaded correctly. Just making a small adjustment to the settings here, and I'm selecting the drop-down option on the right of the deconvolve button, which opens a run batch option. And if I select this, the file open dialog opens, and I need to navigate to the folder of images that I want to deconvolve. So once I've selected the folder, I just need to click process selection, and all the images in the folder will be deconvolved. So now that I've done this, I'm going to show the task manager so we can see the background processes occurring. And here we can see a progress bar displayed for each of my images. This sequence is slightly speeded up. So when Image Pro is finished the deconvolution of all the images, I'm going to navigate to my folder, and there's a new folder being added, and it's a folder called Deconvolved. And inside we see each of our deconvolved image sets, and their names are appended with the number of iterations that was applied in the deconvolution. And here we can see that the image is deconvolved as expected. Thanks very much for joining me for the video. If you have any questions, please get in touch with Media Cybernetics. We'll be very happy to help.